Looking at the span of Chapter 2, we'll be taking a comprehensive look into biological molecules or biomolecules for short. Under subtopic 2.1, we'll be testing for the major biomolecules we have to know how to test. Starting off, the first major class of biological molecules that you have to know how to test for would be your typical reducing sugars. Think of it like the glucose test. Glucose, in fact, belongs to a category of sugars known as reducing sugars. Why? That will be talked about later, but for now, we'll just focus on this. Right, so it's like this. Whenever we have a reducing sugar in a test tube, I'll give you a distinct differentiation of the reducing and non-reducing sugars in a bit. But if you have it in a solution, I mean a test tube, you're going to add a specific solution into this and it's going to give a specific color change. The solution that we're going to add to this specific test for this specific test is known as the Benedict solution. The Benedict solution. The Benedict solution. Benedict solution is actually a mixture of two other solutions, for example, things like copper sulfate. So therefore, I'll tell you what's going on in the underlying process here. First of all, under the Benedict solution, you have blue copper 2 plus ions. We all know the color of copper 2 plus ions is always going to be blue, like so. In copper sulfate solution is going to be reduced. So when we add Benedict solution along, so another constituent of the Benedict solution is going to be the copper sulfate solution. Inside the copper sulfate solution, you're going to have the Cu2 plus blue ions like this. Now, glucose, for example, is a reducing sugar. Example, glucose. It's going to be a reducing sugar. So, it's simply going to reduce the Cu2 plus to reduced to Cu1 plus ions. The color of those things, Cu1 plus ions, are going to be red. That's the underlying principle behind this. If you have a reducing sugar and then you add Benedict solution which contains Cu2 plus ions, these are blue. They are going to turn into a red color if they are reduced. For them to be reduced, you have to have a reducing sugar inside the test tube. If you don't have a reducing sugar, they are not going to get reduced, so there will be no color change. This is known as the Benedict test. We're adding Benedict solution for the Benedict test. The Benedict test. That is possibly one of the worst handwriting. Benedict's test. No case. Hence, the color change that we're going to observe can range from your blue, meaning no reducing sugar, all the way up until maximum reducing sugar. What's the maximum color of reducing sugar going to be? Because it turns red, it's going to turn a very deep red. We typically refer to that as the brick red color. So there's a small gradient, blue to green to a little bit of yellow. Then you're going to get some orange. I'm going to have to move that. And then all the way up until brick red. That's the color change that you're going to observe. Blue means little to none, basically none, little to none, And if you have a brick red precipitate, that's going to be a high concentration of reducing sugar. So as we go along, concentration increases. Concentration increases. Now that's for reducing sugars. What if we have a non-reducing sugar, and what exactly is a non-reducing sugar? Let's see. Non-reducing sugars. Now we have a ton of sugars. Glucose, fructose, galactose, sucrose, lactose. Did I say lactose again? Yeah, I think so. Never mind. All those things are sugars. But most of those sugars that we have already known are reducing sugars. There is only one specific non-reducing sugar you have to know about. Only one. And that is your typical sucrose. Your table salt. Not table salt, table sugar. You call it table sugar. My God. Never mind. Sucrose. The only non-reducing sugar you have to know about. Here's the deal with sucrose. Now, sucrose is actually a disaccharide. Sucrose is made out of glucose and fructose. We'll talk about this constituent under carbohydrates in more detail. For now, just know that sucrose is made out of something known as glucose and fructose. So those things are not capable of producing this color change when combined together. Meaning, sucrose in its sucrose form, in its non-reducing form, is obviously not going to produce this color change. Why? It's not able to reduce these copper 2 plus ions. It's not able to reduce the copper 2 plus ions into Cu1 plus. That's not a possibility. Now here we have to do something else to identify sucrose. I'm going to reduce this in size, keep it here, and here you have to undergo a special procedure known as acid hydrolysis. Acid hydrolysis. What exactly is acid hydrolysis? When it comes to acid hydrolysis, the disaccharide, so our sucrose, is broken down to its monosaccharide forms. To its monosaccharide forms by adding an acid, typically HCl, and then neutralizing it. Once we add an acid, it's acidic, so you use a simple base and then you neutralize it. That is the procedure known as acid hydrolysis. Now what happens when you do acid hydrolysis? It's going to break down into glucose and fructose. Glucose alone and fructose alone are reducing sugars. So now if you put in the Benedict solution, will you get a color change? Yes, this time around you will. This time around you will. The obtained two monosaccharides are reducing sugars. Therefore, they will produce a color change. They will produce a color change. Moving on to starch. 1, 2, 3. This will be the third test we're going to discuss. The starch test. You have known the starch test for a long time. It's the same thing. There's no new information added to it. You simply add a few drops of iodine you have to be specific about what iodine and how we're going to add it. If we take iodine, it's just a solution on its own. We all know that. Iodine is simply a solution on its own. There's no difference. But here you can't just add just base iodine. You have to add iodine in potassium iodide solution. So we're adding iodine. Yes, we are adding iodine. We're adding iodine. But that iodine that we're going to add is going to be in the potassium iodide solution. It's going to be in the potassium iodide solution. Potassium iodide solution. Then if you do have starch present, a positive test gives you blue-black color. You're going to have blue-black color if you do have starch. Moving on to how we're going to test for proteins. The proteins have an interesting test. You call it the Biuret test. So IV, that's for the not Biuret. This is a very common misconception. Okay, don't say Biuret test. It's not Biuret. Biuret is a piece of laboratory equipment. We're not talking about that Biuret. You have to pronounce it like Biuret. You have to pronounce it in the form Biuret. B-I-U-R-E-T. Biuret. Okay. Biuret test. We're using the Biuret test for proteins. Proteins are tested for by the Biuret test. How can we test for proteins and why does it work? If you have a positive Biuret test, let's see what we're going to observe. It's just an empty test tube with proteins right now. We'll see what the color changes are. Proteins have peptide bonds where the amino acids are linked. Peptide bonds with nitrogen atoms. Every peptide bond is going to be composed of nitrogen. It's going to have nitrogen. They form, now you might have wondered why I picked purple to write all of this. That's because they form a purple complex. They form a purple color complex with the Cu2 plus ions that we are adding. So that means that we're adding again something containing Cu2 plus. So once we add that, what's the color we're going to observe? The moment we add that, the color that we're going to observe is going to be blue. So blue means the test is negative, no proteins. But if it does turn into a purple color, then we have a positive Biuret test. What does that mean? We have proteins. We have proteins. If it's a positive test, we're going to change color from blue all the way to purple, and it's a very distinctly nice color change. A very distinctly nice color change, right? I held back a little small thing when it comes to reducing sugars and the Biuret test. Here's the deal. For the Biuret test, heating is not that necessary. Yes, of course you can heat it if you want, but it's not necessary. But for the Benedict test, the reducing sugar test, you need to heat the sample to get the reaction. How do you heat it? Do you hold it to an open flame? Do you put it on the stove or something? No. You have to always use this little piece of equipment known as a water bath and a boiling tube to heat whatever you want in biology. That's how it's done. When it comes to the Biuret test, blue is turned to purple. Alternatively, sometimes potassium hydroxide and copper sulfate separately can be used. Instead of the Biuret solution, we can sometimes use potassium hydroxide and copper sulfate separately to carry out a successful Biuret test. That is a noteworthy mention. Moving forth to the final test, where we are going to be testing for lipids. You can call it the lipid test just, or there's a better term for it. You can call it the lipid emulsion test. It's a better way of saying it. When it comes to the lipid emulsion test, it's very simple. You have to shake the sample. So we're going to have the sample in a test tube or something, or a little bit of lipid. You're gonna shake the sample after adding ethanol. After adding ethanol. Then, if you have lipids, if they are present, will dissolve. Okay? Lipids are not going to dissolve in water. Lipids are going to dissolve in ethanol. So what do you do? What exactly do you do afterwards? After these lipids are dissolved, you have to add this sample, the mixed sample, to a beaker of water. Once you do that, you're going to get a cloudy white color suspension. A cloudy white suspension is formed. Why? Why do you all of a sudden get a white suspension now? If the lipids dissolved previously, now we have water. Now, therefore, the lipids are unable to stay dissolved. They're getting undissolved. That's a word. These are basically all the food tests you have to know under 2.1 testing for biomolecules.